delivering value with integrity, exploring great books worldwide. Hi, this is Sally's Book Club. Today we're diving into the book DNA, The Secret of Life. DNA is practically a household name. Every one of us develops from a single fertilized egg with all the developmental instructions encoded in DNA. This biological macromolecule called deoxyribonucleic acid primarily resides in chromosomes. It not only shaped your development from a fertilized egg, but also connects to those GMO foods we often debate about. Ever wondered how DNA was discovered and studied? This book will guide you in unraveling life's mysteries. As life's genetic code, DNA reveals secrets of aging or disease while tracing species origins. Doesn't that make DNA discoverers extraordinary? Exactly. The 1962 Nobel Prize honored three DNA pioneers, James Watson, Francis Crick, and Maurice Wilkins. Remember these three names, Watson, Crick, and Wilkins. We'll explore their scientific journeys later. The book's lead author is American scientist James Watson, a true life achiever. In 1953, at just 25, he collaborated with British scientist Crick to first model DNA's double helix structure. This breakthrough is hailed as one of the 20th century's greatest achievements, revolutionizing life sciences and our understanding of existence. In 2003, 75-year-old Watson reflected on decades of scientific progress, co-authoring DNA, The Secret of Life with Harvard researcher Barry. Nobody understands this subject better than Watson himself. Through Watson's narrative, you'll witness science reshaping humanity's understanding of life and driving technological revolutions. But more importantly, this book helps you scientifically appreciate biotechnology applications like genomics, DNA forensics, cancer immunotherapy, and genetic screening. This book spans 340,000 words across 13 chapters. I'll explore DNA's secrets through three thematic sections. Part 1, what theories existed about heredity before DNA's discovery? Part 2, how scientists deciphered the genetic code. Part 3, DNA's transformative impact on society. First, let's revisit genetics history. Why do children resemble parents? Since ancient Greece, two main theories explained inheritance. Theory 1, fetuses formed from parental meat buds via intercourse. Theory 2, miniature humans performed in sperm or eggs enlarged during pregnancy. These theories persisted until 19th century experimental challenges. Mendel conducted groundbreaking pea plant experiments during this era. In 1856, Mendel cultivated peas in an Austrian monastery. By analyzing pea traits like flower color, he discovered discrete hereditary units. These paired factors, half from each parent, recombined in offspring, yet their physical basis remained unknown. The mystery was solved 50 years later by American geneticist Morgan. He studied fruit flies instead of peas. Normal flies have red eyes. Rare mutations produced white-eyed flies. Morgan serendipitously found a white-eyed male fly. Surprisingly, only male offspring inherited white eyes. Scientists already knew fruit fly sex linked to visible chromosomes. Morgan's discovery anchored Mendel's theories to physical chromosomes. Since the 18th century, genetics research has captured public interest. However, most people focused on genetics' social implications, believing a parent's behavior and abilities were hereditary. Many then rejected asylum residents, viewing them as degenerate elements threatening racial purity through reproduction. Thus began the notorious eugenics movement. Early 20th century Europe implemented positive eugenics, encouraging reproduction among elites, while America enforced negative eugenics, sterilizing marginalized groups, culminating in horrific consequences. From 1907, 30 U.S. states legalized sterilizing criminals, mentally ill, and feeble-minded. Nazi Germany escalated this into genocide against Jews. Post-War I, eugenics was discredited but cast shadows over genetics research. This concludes Part 1, Humanity's Enduring Quest to Understand Heredity. Since the 19th century, experimental science replaced speculative theories about inheritance. Meanwhile, public misunderstandings fueled eugenic policies. This movement became the final darkness before dawn. Scientific evidence was quietly accumulating during this period. These discoveries enabled Watson's 1953 DNA breakthrough, revealing heredity's true mechanism. Part 2 explores scientists' journey to crack the genetic code through DNA discovery and manipulation. First, let's revisit how DNA's structure was revealed through Watson's perspective. Connecting scientific milestones. Understanding genetic coding requires mentioning Schrödinger's 1944 book, What is Life?, which proposed compressed hereditary information in chromosomes. To understand life, we must first decode its cipher. Schrödinger's book attracted brilliant physicists to study genetic coding. Watson's PhD supervisor and Nobel collaborators Crick and Wilkins were among them. For Watson, this book redirected his career from ornithology to genetic research. But where to begin decoding? Schrödinger suggested chromosomes held the code. Scientists knew chromosomes contained protein and DNA. Most favored protein's complexity. Avery's experiments proved DNA carried genetic information. This discovery electrified Watson. He became convinced DNA was the genetic code. But how to study DNA structure? Many dismissed DNA as too large and irregular for analysis. Watson failed in 2 labs. Fortune struck when he met British physicist Wilkins at a conference. Wilkins had worked on the Manhattan Project, developing atomic bombs. The Hiroshima and Nagasaki bombings shattered his scientific ideals. Schrödinger's book redirected him to genetic coding research. He mastered X-ray crystallography for DNA analysis, a crucial breakthrough. Like airport scanners using X-rays for luggage inspection, Wilkins bombarded DNA crystals with X-rays to reveal structure. Conference images showed DNA's regular patterns, a revelation. Watson realized X-ray diffraction was the key methodology. He relocated to Cambridge University to learn the technique. There, he partnered with Crick and reunited with Wilkins. Within 2 years, they cracked DNA's double helix structure. The DNA double helix resembles a twisted zipper with interlocking teeth. Each tooth consists of chemical structures called bases. With 4 types of bases, the genetic code lies in their sequence arrangement. Genetic inheritance works like zipper replication, copying base pair patterns. How does DNA relate to proteins then? Genetic disease studies revealed DNA-protein correlations, but mechanisms remained unclear. Experiments later identified RNA as the intermediary between DNA and proteins. RNA, another nucleic acid, carries DNA instructions to direct protein production. Crick's central dogma: genetic flow from DNA, right arrow, RNA, right arrow, protein. Next challenge: genetic code manipulation. Scientists began pursuing genetic engineering, playing biological editors. First breakthrough: recombinant DNA technology. Imagine DNA sequences as newspaper pages containing genetic articles. Using molecular scissors to cut desired genes, then glue them onto new carriers. Finally, photocopying modified DNA through bacterial replication. 1960s-70s saw 4 essential DNA tools emerge: restriction enzymes, ligases, plasmids, and bacteria. These tools enabled DNA cutting, pasting, and copying. Too many technical terms. Let's clarify with real-world examples. Restriction enzymes act as molecular scissors, cutting DNA at specific sites. Next are plasmids, circular DNA in bacteria, providing antibiotic resistance. Innovative analogy: scientists realized plasmids could serve as blank canvases with bacteria as copiers. They spliced DNA fragments into plasmids for bacterial replication. Thus, bacteria became recombinant DNA factories. 1973, recombinant DNA technology enabled creating new genetic combinations. Scientists gained biological puzzle-design capabilities. Example: pest-resistant GMO crops, reducing pesticide use. Second breakthrough: DNA sequencing technology. Genetic code lies in 4 base chemicals sequence. Gilbert and Sanger developed sequencing, earning 1980 Nobel Prize. Example: synthetic insulin production using gene sequences. Watson's DNA structure revealed nature's elegant simplicity. 4 bases combinations encode vast biological information. Two key technologies emerged: recombination and sequencing. Part 3 explores these technologies' real world applications. Watson admitted underestimating DNA's societal impact. 9 chapters detail DNA applications from medicine to forensics. We'll explore DNA's crucial roles through key applications. First application: DNA as biological ID, like your train ticket ID but encoded at conception. This genetic ID makes everyone unique from fertilization onward. This biological ID has revolutionary uses. 1985, geneticist Jeffries developed DNA fingerprinting. He discovered variable repetitive DNA sequences in human genes. Unique repetition patterns create barcode-like DNA profiles. This method enables personal DNA fingerprint collection. Applications include paternity tests, forensics, and victim identification. UK-US established national DNA databases using this technology. Cold cases get solved through preserved DNA evidence matching. Example, 1973 UK murder case solved via DNA 26 years later. DNAology application. DNA also traces family trees through sequence similarities. Two DNA types: chromosomal DNA and Y chromosome, paternal lineage. Mitochondrial DNA traces maternal ancestry exclusively. 1987 global mitochondrial study identified common maternal ancestor. This conclusion is astonishing, but it's not sensationalism. 2000 Stanford study confirmed 150,000 year human lineage via Y chromosome analysis. Second application: DNA in disease prevention and treatment. Many genetic pathology bases stem from specific gene mutations. Traditional amniocentesis carries 1% miscarriage risk and late-stage detection limitations. Early embryonic genetic screening could solve these dilemmas. British physician Winston pioneered this breakthrough. 1990, first successful preimplantation genetic diagnosis, PGD. Screening IVF embryos for mutations before implantation. This technology is now available in China. 2014 Peking University third hospital case. Healthy babies born using single-cell sequencing. DNA testing also identifies cancer-related gene mutations. Example, 1990 BRCA1 gene discovery in breast cancer families. BRCA1 mutations increase cancer risk to 80% by age 50. Preventive measures for mutation carriers reduce cancer risks. 2013 Angelina Jolie reduced breast cancer risk from 85% to 5% through preventive surgery. Crucial note: preventive surgery isn't foolproof, cancer risks remain. Beyond prevention, DNA modification enables cancer treatment breakthroughs. We can genetically engineer immune cells to better target cancer. RT therapy, extract, modify, reinfuse immune cells to fight leukemia. 2013, KRT saved Emily Whitehead from terminal leukemia. 27 FDA approval marked new era in cancer treatment. Now shifting focus. Third application: GMO crops through recombinant DNA tech. Traditional pesticides contaminate food chains and ecosystems. 1862's Silent Spring exposed pesticide dangers globally. Genetic engineering created pest-resistant crops. Example, Bacillus thuringiensis, Bt bacteria's insect-killing protein. BTA toxin only activates in alkaline insect guts, safe for humans. Inserting Bt genes into crops creates natural pest resistance. Insects die after nibbling, mammals digest safely. DT crops now widely adopted: corn, cotton, soybeans, etc. Genetic modification boosts crop yields while reducing pesticide use. Yet some Americans irrationally fear GM tech as dangerous monsters. GM crops. Post-launch protests erupted across U.S. against biotechnology. Watson condemns anti-GMO fear-mongering while ignoring benefits as absurd. Opposition often stems from emotion rather than science. He counters with several arguments. Argument 1: GM crops are unnatural. Modern crops differ drastically from wild ancestors through hybridization. Modern wheat evolved from einkorn wheat and goatgrass hybrids. This too constitutes genetic modification. Argument 2: GM foods are toxic, yet plants naturally produce defensive chemicals. Natural versus engineered toxins share similar biochemical properties. Difference lies in origin: natural selection versus design safety. Bt protein example: lethal to insects, harmless to humans. Watson stresses China's need for GM crops to feed 1.4 billion people. China will pragmatically advance GM research to ensure food security. Western hesitation may cede biotech leadership to China, though current Chinese debates prove complexities persist. We've explored DNA's crucial applications through three lenses, from forensic IDs to medical breakthroughs and ancestral tracing. By editing this genetic code, we engineer pest resistant supercrops. Notably, DNA, The Secret of Life, was published in 2002. The past 2 decades witnessed explosive progress in both research and applications. CRISPR gene editing and cancer immunotherapy now lead new medical frontiers. Immunotherapy transforms cancer into manageable chronic conditions. A dawn of hope. This book unveils life science's grand tapestry, from forensics to transgenic agriculture. Biological innovation maintains relentless momentum today. This century-old scientific revolution will profoundly reshape humanity's future. And so concludes DNA, The Secret of Life. Find detailed notes below. Until next literary journey, happy learning.